sort of the basis structure, the building blocks of proteins are amino acids. So then the question of, okay, so we talked about like polar amino acids, non-polar amino acids, ionic amino acids. So you don't have to know the names of them, but you should know that there's different types. And remember that it's based on that R group. So it's based on the group that's different amongst those 20 amino acids. So there are 20 amino acids that are used to build proteins in the human body. And out of those 20, 8 of them are called essential. Essential amino acids you have to get in your diet because your body can't make them. The other 12 you can make from other things. You can make them from lipids, you can make them from other amino acids, you can make them, build them from carbohydrates, so you can build them from other things, but 8 of them, you have to eat these. That is what they mean when they say that they are an essential amino acid. They have to be eaten. If you don't get these regularly in your diet, then you may end up being protein deficient. If you don't have the building blocks, then you can't build the proteins that your cells need. And if your cells can't build the proteins that they need, then you're going to lose muscle mass. You are going to have a weak immune system. You're not going to be able to build the proteins that the body needs. A complete protein in your diet is one that has all the essential amino acids. And remember, any kind of animal meat source, so that is beef, chicken, pork, fish, as well as eggs and milk, those are considered complete protein sources because they have the essential amino acids. And remember that strict vegetarians, they don't end up being protein deficient because they eat combinations of grains and legumes, which are beans, and those, between the two, they can get all 8 of the amino acids that they need, or 8 of the 8 essential amino acids they have to have. So then we did go on and start talking about, okay, so what is protein structure? So yes, you have amino acids, they're the building blocks. What link them together is through a condensation reaction. So remember that a condensation reaction, that is when you form water by pulling a hydrogen or pulling 2 hydrogens off of molecules and an oxygen. So in this one, the carbon side of the amino acid loses an oxygen. The nitrogen side of the other amino acid loses 2 hydrogens. So that's where your H2O comes from. And that forms the link between the 2 amino acids. So you can see this link here, it's called a peptide bond. So this creates a chain of amino acids. So one amino acid linked to another one, linked to another one, linked to another one. And so we did that as an example. So this is amino acid. There's amino acid 1 linked to amino acid 2, linked to amino acid 3, linked to amino acid 4. Every single one, you pull off 2 hydrogens and an oxygen, 2 hydrogens and an oxygen, 2 hydrogens and an oxygen, in order to form the peptide bond, which is this link that holds those amino acids together as a chain. So that's really the big thing to remember for what they call the primary structure of a protein. It is the order of the amino acids in a chain linked together by a peptide bond. So an example of problems that can come about by just these protein chains is what happens with somebody that has celiac disease. So somebody that develops celiac disease actually has an allergy. They have an immune response to a protein called gluten. So gluten has really been talked about a lot in the last 10 years. People are like, oh, gluten's bad. And then you're like, well, what is gluten? I don't know. It's just gluten. Gluten is actually a protein that is found in wheat. Most grains, so it's not found in rice. It is not found in oats. But it is found in like barley, it is found in wheat, it is found in like all the different kinds of ancient grains like buckwheat and those kinds of things. So this protein, when you eat it, it gets digested. Digestion starts in your stomach with proteins. So you end up with these little pieces that are in the small intestine. These pieces of this gluten protein are seen as foreign, just like If you have any other type of allergy, if you have a food allergy, your body responds because it thinks that it's something that's foreign. So the same thing happens, but these protein fragments create an inflammatory immune response in the small intestine. So the person's small intestine begins to swell. The lining gets inflamed. They have a lot of pain. They may have a lot of gas, bloating, nausea that goes along with it. But they can also have malabsorption of nutrients, which means they're not getting the nutrients that they're eating. And oftentimes, because of this flare-up that they have, they really don't want to eat anything because it's like everything they eat creates this pain because the lining is inflamed. So even if you're eating something without gluten, it's sliding by this really swollen lining. So what actually happens over time, somebody that has celiac disease, the lining of the small intestine actually begins to shrink. So when looking at this, you see the villi of the small intestine. So that's this sort of like this little cartoon here. If you think of your small intestine microscopically, it's got all of these little finger-like projections, kind of like a thick pile rug. And so food and nutrients slide around through these finger-like villi projections, and that, when they hit the wall of the small intestine, that helps them to get absorbed. But if you lose the villi, which is what happens with celiac disease, so you see it's like basically flat. So nutrients pass through and never even hit the wall of the small intestine. So that is why you end up having very poor absorption. So low intestinal absorption occurs. So I've had two friends that have celiac disease, and one of them, the first time that they had it, they were just constantly complaining about pain in their abdomen. Like they were just got to the point that they really weren't eating very much. They lost like 30, 40 pounds. They go to the doctor, and the doctor's like, well, clearly there's something wrong. So they start doing tests. So they have to end up doing an upper GI, which shows this inflamed small intestine. Testing shows that they were actually having loss of the villi. Put them on steroids, first of all, because you've got to get that inflammation down because the lining is so inflamed. So you put them on steroids to try and calm down the lining, and then take gluten out of the diet, which means now you can't eat anything with bread. There's no bread, no cereal, no crackers, no pasta. All of those things are wheat containing. There are now like different gluten-free types of noodles that they make and breads that they make. But that's the big limitation, is they've got to cut out all of the wheat-containing foods in the diet. And then slowly over time, they were able to go back to normal diet, like normal absorption of nutrients. They basically just can't eat anything that's wheat or wheat-based for the rest of their lives. And the other person that I knew, he was just always stick skinny, and I never really understood why. And I was like, why are you like, you're like this little beanpole? And he's like, oh, I have celiac disease. And he was like there's such a limit on the things that I eat. Like I get really tired of eating rice all the time. Like I can eat rice and I can eat potatoes. And pretty much that's the only starchy kind of food that I can eat because all of these other grains are all an issue. And he even had symptoms if he ate oatmeal that was processed in a factory that processed wheat, he would end up having inflammation. So like he just like completely cut out any kind of grain because he found like even little amounts of contaminants in the foods that he was eating would trigger that too. So he like basically didn't go out to eat very often because even if he thought something was gluten-free, that wouldn't necessarily mean that it was the case. So he was really, really careful about what he ate because of how bad it would get if he had anything that had a wheat. But this is just from a protein, and it's purely because of the protein sequence. So having those sequences causes this immune response to occur. Well, proteins aren't just a straight chain. Proteins take bends, they curve, they coil. Two of the kinds of coils that you can see, they call this secondary structure because proteins aren't just like a string. Instead, they take on what's called an alpha helix shape. Can you kind of see how this looks like a coil? So the alpha helix structure is because of hydrogen bonding of the backbone, that nitrogen, carbon, carbon, nitrogen, carbon, carbon backbone that is in the protein chain, not the R group, but the actual part that ends up being linked into the protein chain itself. They can hydrogen bond. So the nitrogens can hydrogen bond with the oxygens of the carbon, and that creates this kind of like spring. So can you see that if you had this straight chain and you pulled on it, it might snap. But what do you know about a coil? If you take and pull on it, what does it do? It'll stretch, right? So it's got this flexibility. So this really helps to give some stretch and recoil. So the protein is not as easy to break because it's kind of like a slinky. So you can end up pulling on it and it'll bounce back into its shape. It's going to give it a lot more flexibility. This is much stronger than if you had just a straight chain. If it didn't have this coiling, it'd be a lot easier to snap it. The second type of secondary structure, it looks like a fold, I remember making fans. If you took a piece of paper and folded it back and forth and back and forth. So you would get what they call a pleat. So they say it's a pleated sheet. This again is folding of the protein that kind of makes these big zigzags, but they hydrogen bond to hold them in place. And so again, if you think about like a folded piece of paper, you could push it and you could pull it. So it's got this flexibility to it. That also helps to stabilize the structure of the protein. Proteins don't tear very easily. They don't snap because this is going to give some flexibility. So they call this one a pleated sheet. So helix and pleated sheets are ways to give the protein a little more flexibility. Example for this, changes in secondary structure in brain proteins is what is associated with Alzheimer's. So what happens with the person that has Alzheimer's is they become more and more forgetful initially. So they may like look at you and be like, I know you, but I don't know you. Their short-term memory it gets worse. Long-term memory, they start remembering things that happened 25 years ago, but not things that happened like an hour ago. And then they begin to lose that, and you begin to notice that their actual function begins to sort of go backwards. They start acting more and more childlike over time. They then end up losing like their ability to controlling their bowels. They can't remember how to do things like eat by themselves. So it really kind of like progresses. They have done research that has shown it is because of changes in the coiled nature of proteins that are found in neurons. So here, normal patients, their proteins in their neurons look like helixes. So these are those alpha helixes. But they have found that the neurons in the brain for patients that have Alzheimer's, there is a change in the folding of the proteins. So instead of being helixes, they look like pleated sheets. But that pleated sheet arrangement causes those neurons to deteriorate and form what they call plaque. So basically, it's like scar tissue. And so this is why the Alzheimer patient, it starts off and then progresses over time, having less and less brain function, which leads to all of those characteristics. So they have actually identified changes on a protein level of what happens in somebody's brain as Alzheimer's progresses. So now there's a lot of research into why. Like, why do we have this shift in the structure folding? Why does this secondary structure change? Is it completely genetic? How come it was normal and now it's changed? And working with medications that help to block this change, helping to slow the progression of Alzheimer's over time. So the third level of structure is really all based on those R groups. So remember that we said that you have the hydrophobic R groups, those non-polar R groups. We have hydrophilic R groups, those that are polar and those that are ionic. They are going to end up changing and affecting those alpha helixes and those pleated sheets because things that are nonpolar do not want to mix with things that are polar. So the nonpolar side chains, they want to clump together and form little clusters. Polar groups, they want to be out near the water. So polar groups like to be on the surface of a protein. So if it's bent in, but it's polar, then it might cause the whole protein to have a turn, to shift, and allow it to interact with other polar groups or water because it can hydrogen bond. Remember the charged R groups were ones that had positive or negative charges on their R group. They like to come together and interact much like salts do. So they call those salt bridges. So if you have like a positive nitrogen and a negative oxygen, they are going to be attracted to each other. This creates what's called tertiary structure. So tertiary structure is all because of the R group. Here's a good picture. So this purple string, this is a protein. Okay, so the string, the order of the amino acids, that's the primary structure. Do you see some helix in here? Do you see some coiling? Do you see where it folds back into a pleated sheet? That's secondary structure. But notice that's not all there is. It has these extra folds that are stabilized because of tertiary structure. So in this one, notice that you've got some hydrophobic R groups. So notice those are like those aromatic groups. There's just a CH3. Here's a CH3. They want to all sit together because they're all carbon, right? They don't have anything polar. They're non-polar groups. So they're going to cluster together. Here's an example of a salt bridge. See that positive nitrogen and the negative oxygen? They're attracted to each other like magnets. So they're going to form this link. So instead of that area to here with the salt bridge being like a long coil, it's like a long coil that folds over and stays now in this folded double coil shape. Tertiary structure is the reason that proteins form a three-dimensional structure that leads to what they call a globular protein. So when I think of a glob of anything, I think of like something that's like more rounded, more spherical in structure. So globular proteins have lots of tertiary structure. They're going to have lots of extra folding. An example of a molecule, a protein that has a lot of tertiary structure is going to be proteins or enzymes. And it's enzymes because enzymes have a certain shape that allow them to interact with reactants, bringing them together to help speed up chemical reactions. Things that have a lot of folding are going to have a lot of tertiary structure. So here you can see some examples. The first one, example, enzymes. I just say a lot of cellular proteins are globular proteins. But do you notice that kind of gold? You can see the coiling. You can see how it kind of folds back in on itself. But this has a much more three-dimensional shape. That's a globular protein. Whereas the ones down at the bottom, there are a lot of proteins in the body they call fibrous proteins. So fibrous proteins, they have primary and secondary structure, but they don't have a lot of tertiary structure. Fibrous proteins look stringy. So they just form long alpha helixes. And those alpha helixes don't fold back on themselves. So fibrous proteins are going to be much more string or rope-like. Fibrous proteins tend to not be soluble in water, but these are really good anchors. Collagen, the most abundant protein in the body, is a fibrous protein. Collagen is the ropes that form tendons, ligaments, the anchors that helps to form all of what they call connective tissue. So like loose and dense connective tissues that help to hold layers together. All of your organs have a wrapping on them. All that's connective tissue. Those are all fibrous in nature. These are nice and durable, strong. They show collagen. They also show keratin in this one. That's the first one. So keratin is what makes up your skin, your hair, your nails, right? So these are insoluble in water. So that way things don't just wash away. So that helps to hold everything together. So keratin is the other one, the waterproofing protein that covers the surface of the body. These are all fibrous. Just think of fibrous proteins. They don't have a whole lot of folding. They're much more stringy or rope-like, and they help to hold stuff together. Globular proteins have a big folded three-dimensional shape, and their shape is the reason they do what they do, like an enzyme, right? The shape of the enzyme is the reason that it can interact with certain things. So looking at this, these amino acids, and there's 1, 2, 3, 4, 5, 6, 7, 8. There's 8 of them. So if you look at this first one, would you think that this first one, would you describe this as being hydrophilic or hydrophobic? It's hydrophilic because of the oxygens. It's a polar. It's this part is the R group. This is the part that's going to want to interact with water or interact with other polar groups. What about this next one? So we see the charge on it. So that charge, that one right there, that's what's going to make this ionic. So this is also hydrophilic. But what about the next one? So you notice this is nothing but carbon and hydrogen. So this R group is nonpolar. So if it's nonpolar, it's going to be hydrophobic. Right? Nonpolar, nothing but carbon and hydrogen. So what about this next one? So the next one's got an aromatic ring, but it's got an OH on the end. What do you think of this one? It's hydrophilic. So this one's polar. What about the next one? There's a CH, CH3, CH2, CH3. That's a hydrophobic. So this one's a hydrophobic because it's nonpolar. The next one's a nitrogen with a positive charge. Yeah, this one's ionic. And ionic, because it's got that plus positive charge, ionic, remember, is hydrophilic. This one's an odd-looking one because this is a carbon, this is a carbon, this is a carbon. And it's part of the ring. It's like kind of like loops back on itself, but it's nothing but CH2s. So that means it is hydrophobic. So this is going to be a nonpolar. And what about the last one? What do you think of the last one? It's a CH2 with an aromatic ring on it, which is 6 carbons and hydrogen. It's going to be nonpolar as well. So if we had this protein, we might get an alpha helix twist to it, but the things that are hydrophilic, they're going to want to either be on the outside of the protein or they're going to be close to each other. They're not going to want to be tucked in and be close to the nonpolar. But then those nonpolar ones, and we've got 4 of them, these 4, which are not side by side, well, 2 of them are, but they're not side by side, but they're going to cause the protein to shift so that they can tuck together. So those nonpolar are going to try to tuck themselves inside of the protein, kind of like the soap molecule, right? They're going to want to try to tuck themselves in, and the polar or hydrophilic are going to want to be towards the outside. So that's going to create tertiary structure, that folding. So if all your amino acids were polar, you would have a more fibrous protein, right? So if all of your amino acids liked water, then that means that they would all be towards the outside, and you would end up with something like collagen. Adding those non-polar amino acids creates that folding need to try and keep them dissolved. Fourth level of structure. So the first level of structure is just the sequence of the amino acids. Second level of structure is the helix or pleated sheet that the chain makes. Third level of structure is the polar-non-polar interaction, hydrophilic and hydrophobic. The last one is quaternary structure. So quaternary just means like the fourth level of structure. This is if you have two or more proteins. So if you have more than one protein, then those proteins have to fit together. Some proteins don't have quaternary structure. The only way they have quaternary structure is if you have more than one protein chain that come together. Now, I'll tell you that collagen and keratin, which are fibrous, if you look at that down at the bottom, do you see that it's more than one chain? I would just be able to magnify it. So there's collagen and keratin. I'll try to hold them. So do you see how it looks like thread that's twisted together? So this actually has quaternary structure. It doesn't have tertiary structure because it's more thread-like or fibrous. So there are 3 chains for collagen, 2 chains for keratin. But they have quaternary structure because there's more than one chain, more than one protein. And these ones look like they're woven together, like into a rope. And so 1 is a whole lot better than, 3 chains is a whole lot better than just 1 in terms of strength. In fact, if you look at thread that you sew with, it really looks like collagen. So if you look at thread up close with like a magnifying glass, you would see that it is actually threads that are woven together in order to make a strong type of anchor to be able to link things together. So those, even though they're fibrous, they actually have a quaternary structure just because they have more than one protein chain. Here's another example. So in this one, this is showing hemoglobin. So here's the primary structure, right? We said it's just the chain, the order of the amino acids. Secondary structure, this is the coiling. Either coiling or pleated sheets of the chain. So it's just the backbone of the protein chain that does the secondary structure. Tertiary structure is what happens when you have folding. So here you have hydrophilic, hydrophobic that creates that folded nature. Not all proteins have tertiary structure, but if they have a lot of hydrophilic and hydrophobic amino acids, they do. But then this last one. So this last one actually shows hemoglobin. There are 4 proteins. There's 2 that are colored in gray, 2 that are colored in purple in this image. Notice that all 4 of them have a lot of tertiary structure. They're kind of globular looking. One protein can carry oxygen, but not very well. But when all 4 of them come together, it changes the shape of each of those protein chains and makes them able to carry oxygen very well. Your red blood cells are full of hemoglobin. Do you notice also in there, do you see those little red things? I don't know if you can see them. Not that it'll let it blow up, but do you see those little like red things with a little yellow dot? What does a red blood cell have that makes it special? It's got hemoglobin. It's really nothing but hemoglobin. Like 98% of a red blood cell is this protein. But what does this protein have? It has 4 of those. It's the reason that a red blood cell is red. What's inside? Iron. Each of those little subunits holds an iron atom. That is the reason why a red blood cell looks red, is because of the iron that is contained within. And this structure actually can hold 4 oxygens per molecule. So each of those irons can hold an oxygen. So you have 4 oxygens for 1 hemoglobin. And every red blood cell has about 1 million hemoglobin proteins. So that means 1 red blood cell can carry 4 million oxygen atoms. If you don't have iron, you have a reduced oxygen carrying ability. If the hemoglobin doesn't have the right shape, it lowers its oxygen carrying ability. This is what happens for people that have sickle cell. So in sickle cell, the hemoglobin protein does not have the correct shape, and that lowers the amount of oxygen it can carry. Globular proteins, that folded up nature, is really sensitive to changes in its environment. So if you take a globular protein and you shake it, or you heat it, or freeze it, if you add acid or base, if you add heavy metals like lead or mercury, this can take this folded up looking protein and cause it to unfold. It'll lose its quaternary, tertiary, and secondary structure. And if it does that, it may not be able to fold back up. So this is especially true with a globular protein. Globular proteins are much more sensitive to being denatured. Because they have a lot of secondary, tertiary, and quaternary structure. Fibrous proteins like collagen, they're kind of thread-like. So they might start to unwind, unravel, but they're not going to unfold as quickly as a globular protein will. So examples of this. So in terms of shaking, so if you take egg whites and you just start to whip them, okay? So that is by mechanical force, by shaking that, what happens? It goes from being this gooey kind of off, like ivory colored liquid to being what? So you get out a mixer, an egg white, and you just turn it on high. What do you end up making? It gets white, it gets fluffy, you make meringue. Okay, so meringue is actually egg white protein that has been shaken so violently that it has actually caused those proteins to unfold. It goes from being a liquid to being this big white fluffy solid. Heating and freezing, they're showing what happens when you fry an egg. So if you take an egg and put it on in a fry pan, that heat, you see that jelly-like kind of egg white begins to turn white. You leave it there long enough, it'll become solid. That's denaturing it because of heating. So you're taking something with all this folded nature and heating it just causes it to unfold. And when it unfolds, it becomes solid. And does the egg white, like if you put it on a plate, will it reform? No, it stays solid and white. So denaturation is permanent. Example, lead paint. So way back when they developed paint, they found adding a little bit of lead to the paint helped the paint stick to the walls better. This was really helpful in like trim and outdoor, like if you're painting the outside of your house, because this meant like the paint would be good for like 20 years. You did not have to repaint every couple of years. So the lead really helped that paint to adhere. The problem comes in is that lead, when ingested, it does not get filtered out by your kidneys. It gets absorbed and it can end up collecting in the brain. So in children especially, with their growing brain and neurons, they are more subject to damage because of denaturation of proteins in the brain. So what they found was kids that were exposed to lead-based paint, that they would have more reading difficulties, more learning difficulties, more focusing type of difficulties all along. And if it wasn't remedied, it became permanent because I said denaturation's a permanent thing. So you're like, well, why would kids get lead? Okay, so one, if you have a house that's 100 years or older, then odds are there is lead-based paint somewhere in that house. And so when the lead paint does peel, like think about like the trim around a window inside. So little kids in their room playing, and here's this little bit of paint that's starting to chip. What do kids like to do? Pick at it, and then what are they going to put it? In their mouth. Lead-based paint actually tastes sweet. The lead in the paint can trigger a sweet taste. So if a kid takes a little piece of paint chip and it's a little sweet, what are they going to do? Keep eating it. Yeah, let me just pick this all the way off. Nom, nom, nom. Yeah, this is like having like little tiny candy bits. That's what they found out. Because they found that this lead levels, as that went up, that you had learning, development, neural kind of expansion decreases, and the fact that it has a little bit of a sweet taste made it a bigger issue. So that is why in the 1940s, they outlawed lead-based paint. Problem comes in getting rid of lead-based paint, because if you go and sand it, now you have lead paint as a dust. And so now you're inhaling it. So you're still going to end up like increasing the amount in the body. So now you either like leave and they bring in like a crew that cleans everything up and like sands everything down. And you have to have special ventilation, and you have to like make sure that the cleanup is extreme. They have to like go and swab and do lead paint swabs all around to make sure they get it. Or you like primer it. So you like paint over it so that it is like sealed within. So it's sort of a one or the other. Some people don't like the idea that it's still there, but sometimes it can be cost prohibitive to try and remove it. So lead, mercury, another one. They found that some people had issues with working in the chrome industry, working in the with a lot of metals, all because of the presence of heavy metals that can cause that. So these are all examples. Heating it, adding an acid or a base. Adding acid or a base changes the hydrogen ion content, and that can change how that tertiary structures interact. Detergents, soaps. So soaps are really good at being able to dissolve and break up membranes. They can break up membrane proteins as well. And then the last one we were talking about was like shaking, mechanical agitation. I always just think about like beating egg whites until it becomes meringue. So this leads us into the sort of like last half. These are the protein functions. So we've kind of like mentioned all these proteins as we've talked about this job, that job, blah, blah, blah. But we can really say that proteins have 8 overall functions. So we'll list each one and then give you an example. The first 3 are actually all together. So the first 3 are all on one slide. So proteins, they can be messengers, receptors, or transporters. Messengers, think hormones. Insulin is a really good example. So we've talked about insulin, we've talked about diabetes, we've talked about the importance of insulin. So remember here, insulin, and they show it down in the bottom image, insulin is the little yellow ball, like the little globular protein coming along. Insulin binds to a receptor, And when insulin binds to a receptor, that opens glucose channels, right? So you have to have insulin, then blood sugar can go into the cell. If insulin's not there, that glucose channel stays closed. So that's why insulin, if blood sugar increases, insulin's there, helps to bring glucose into the cell. What insulin binds is a receptor. Receptors are proteins. Receptors have a very specific shape. That insulin receptor only binds insulin. So if another hormone comes along, it has no effect, like growth hormone or thyroid hormone. It is only specific to the shape of insulin. So these are good examples of where that shape of one protein interacting with the other. And then the third one is the transporter, or you could think of it as channels. We talked about these when we talked about the cell membrane. So we talked about how you have phospholipids as the bilayer, that's the barrier, but then you've got these integral proteins that can act as a channel, and they're going to let things go in or out of the cell based on what that substance is. So there's all three. The messenger are hormones, the receptors are what the hormone binds to, and the transporter is the channel that allows molecules to move in or out of the cell. All of those are proteins. If you don't eat enough protein, you can't make those. Then you might not be able to make insulin. You might not have enough transporters. You might not have enough receptors. So that is why having proteins in your diet enables your cells to build the things it needs to build. Okay, so those are three. So there's only 8. So this is 3, all on one. Just like remember all 3 of them together, right? So remember, like the messenger, the hormone, binds to a receptor and causes a channel to open. All 3 of those are proteins. 4, carriers. We talked about this one, hemoglobin. Okay, hemoglobin is a carrier, and its job is to carry oxygen. It has 4 protein chains, so it has quaternary structure. This picture, they color it so that there's the 2 blue chains and the 2 red chains. The other one was purple and gray, but they're the same. So remember that each hemoglobin has an iron, so it can carry oxygen. Other examples of carriers that we talked about. Remember LDL and HDL? That stands for high-density lipoproteins and low-density lipoproteins. These are carriers of what? What do you think when you think HDL and LDL? You think about cholesterol, because that's their job, right? So their job is to transport fats and cholesterol through the blood. So you can remember that as an example. So think of hemoglobin as a carrier of oxygen. HDL and LDL, they're carriers of cholesterol, and they're all proteins. 5, protection. Good example of protection, antibodies. So antibodies are made by immune cells, or made by B cells, which are a kind of lymphocyte. These proteins bind to things that are foreign. So they recognize, attach to things that are foreign, and that helps to trigger an immune response. So example, what? I don't even know what day I was out. But I had COVID, okay? So by being exposed, now I have this virus that has infected my airways, has infected all of for the most part, it was all upper respiratory kind of stuff that went on. Triggered an immune response. I had a low-grade fever, had a headache, I was really tired, I had a little bit of a cough, my nose was running, all of those symptoms, those are all responses to something that's foreign, okay? So if you have like any kind of a respiratory virus, all the symptoms you have are because the body is going, whoa, something's here. Like it's all those symptoms are triggered because something is there and it's causing irritation. At the same time, your immune system cells are hunting for it. Your immune system cells are looking for cells that have those virus particles inside. And when they find them, they start to build antibodies. And what an antibody does, so here's a It could be a foreign cell or it could be a virus-infected cell. Either one. If you have a cell that's infected by a virus, that cell is now considered foreign. If it's a foreign cell, these antibodies have the ability to stick to the cell. And I'm not saying like one or two. We're talking like millions. So this cell is going to have millions of little antibodies attached to it all over. Antibodies are a signal that there's something foreign. So if you look at that cell, which used to be sneaky, it used to be able to just go along from point A to point B. Now it has 1 million antibodies on its surface. Can it like hide? No. This literally marks that cell as foreign, and it brings immune cells like macrophages in to attack it and destroy it. So that's really what an antibody does. And it's specific. So it actually can recognize only cells that are infected, only cells that are foreign. They're that specific. So now, hopefully, if I'm exposed to this again in the next, I don't know, until there's a new mutation, if I'm exposed to the COVID virus again, I have those antibodies already circulating in my blood. And I have B cells that can recognize and immediately start making millions of antibodies. So instead of those virus particles spreading and spreading and spreading, what happens? They get stopped. They get attacked and removed before they make you sick. Have you ever and I was hoping this is what it was, like the night when I got sick, I was like, I just don't feel right. Like that sensation. Have you ever had I feel like my glands are swollen. I just feel kind of crummy. And you wake up the next day and you're fine. Have you had that happen? Okay, that's because your immune cells kicked it. Your immune cells did this. They got rid of whatever was starting to make you sick, so you really didn't get it. You might have felt like something was off. I had that feeling on Monday night, and then Tuesday I was like, oh, no, I feel worse. So I knew that it wasn't something I was just going to be able to get past it. But that exposure, now my immune cells can make antibodies to foreign COVID-infected cells so that they can respond fast enough to help protect me, clearing it before it actually causes all those symptoms again. So that's how, if you've had chickenpox, that's why you don't get chickenpox. Even though you might be hanging around with people that end up with chickenpox, you don't get chickenpox again because you have these antibodies. So that's how they're a protection. 6, contraction. I said almost all of your proteins that you ingest, the vast majority of your protein comes from meat. Meat is skeletal muscle. 80% of the inside of a skeletal muscle cell is protein. So it is a very high protein content cell. Actin and myosin are the 2 muscle proteins that allow you to move. So in contraction, the little thin, the ones that I drew kind of in blue, the little thin lines, those are the ones they call actin. They're the thin proteins. And the big, thick-looking red ones, those are myosin. And they're kind of overlapped loose when your muscle is relaxed. But when your muscle contracts, you know that it shortens. And so what happens is the actin actually the myosin pulls on the actin and creates an overlap between the two. So they pull on them, creating this overlap. And that causes the entire cell to shorten. So there are millions and millions, like in the billions, of muscle proteins that fill your skeletal muscle. And using energy, they can allow for contraction. So if you get like a steak, you get chicken, you have pork, meat, pork chops, whatever, all that is basically cells that are filled with actin and myosin, and that's your protein source. But as animals, we use that for contraction as well. So that's 6. So we have protection, contraction, anchoring. We've actually already talked about this one. Collagen. Collagen, I said, is the most abundant protein in the body. It looks like a thread, okay? It is like 3 proteins that are just wrapped to make this thread-like protein. It's what your tendons are made of. It's what your bones are made of. Cartilage is made of collagen. Ligaments are made of collagen. So all of those, all your connective tissues, all those fall into that connective tissue. This is the primary connective tissue that you find, and it helps to hold things together. So it's like what anchors your muscles to your bones. It's what helps bones to bones. It helps to hold your skin on, okay? All of that connective tissue that holds layers together. An interesting one that's another protein that has this job of anchoring is a protein called elastin. So elastin is like collagen, except that it has more beta pleated sheet. So see how it sort of folds back on itself? Well, you can take elastin and you can pull it, and when you let go, it bounces back. So this is why you can like pull your skin and it goes back into place because of this elasticity that the skin has, but there's actually a protein that holds it that way. When you get older, this stretches and it doesn't bounce back, and that's what happens when you get wrinkles. Exposure to sunlight, smoke, those types of things, not enough water, all those will help to cause elastin to lose its stretchiness over time. But those are 2 important anchors. Collagen's really the biggest one. And then the last. So the last one's enzymes. Okay? And we've talked about enzymes. So what you really want to remember is that enzymes are catalysts. They speed up chemical reactions. They don't get used up. So remember that in this, an enzyme can interact with a reactant, speed up the reaction, release the products, and then go do it again. So they can do this over and over and over again. The structure of this, like the one that I gave you, was in the reaction where you have A and B, and you want to make C. And the enzyme has this shape, so that the shape of A and the shape of B is specific so that they fit into the little clefts of the enzyme. So A and B. Once they bind to that enzyme, the enzyme can then bring them together. So now A and B are side by side. So remember, we said that in order for reactions to occur, things have to come in contact. Well, look, they come in contact. And they have to come in contact in the right orientation. Remember, I told you that sometimes, like, if reactants bind this way or they come together this way, nothing happens. They have to come together in the right position, too. Well, enzymes are going to put them together in the right position so that the reaction speeds up. It doesn't require as much energy. The activation energy lowers. And then you end up with the product. But the neat thing about enzymes is that enzymes can now do this again. So an enzyme doesn't just do this once. An enzyme can do this over and over and over and over again until it basically wears out. They sometimes call the reactants the substrate. So just know that. If you see the term substrate, just remember that's the reactants. It's the starting material in the reaction. So here, like glucose and ATP, those things are the reactants, but I would also call these the substrates. So don't let that throw you, because sometimes that's people are like, So I told you in looking at this one, looking at the energy, the amount of energy needed if I don't have a catalyst, that's the activation energy, right? Would be this height. But if I add an enzyme, my activation energy is going to be much lower. Because enzymes bring things together in the right orientation, and that makes it easier to form or break a bond. And maybe you remember the image that we saw in chapter 5, I think it was. Do you remember that E had like the little special shapes? So very specific shape that it interacts with. Things that affect how fast an enzyme works. One is starting material. So remember I said substrate is like the reactants. So if you have a lot of starting material, then the enzyme can take and do this and take and do this. It can do this reaction as long as it's got something to use. So their analogy that they use is these three guys that are building bicycles. So if you didn't have enough parts, they couldn't make as many bicycles, but it says their maximum output per day would be 25 bicycles. So if you had 50 parts for 50 bicycles, they're still only going to make 25. But if you don't have enough parts for 25, then they're only going to make what they can. So think about it's like starting material. As long as an enzyme has plenty of starting material, it's going to go as fast as it can. There is like a maximum point. So they do have like kind of, it's just as fast as they can work, right? You can't do things but so fast. Enzymes are the same way. So they sort of have like a maximum rate. If I change the pH, if I add acid or base, that can change the shape of the enzyme. If I change the shape, then it might not bind to the substrate and it might not speed the reaction up as much as it should. So changes in pH can change the enzyme shape. And if you change the shape, now the substrate might not be able to bind. There's a couple of examples of enzymes here. Notice that most of these enzymes work well about pH of 7. Well, that's neutral. That's more like body pH. Notice like urease, pH of 7.4. That's the blood's pH. Okay? But pepsin is in the stomach, and it has evolved to only work under very acidic conditions. So when the pH in the stomach is 1 or 2, pepsin works great. If you took too much antacid and your pH rose, your pepsin may not work as well. It may not be able to digest those proteins because it doesn't have enough acidity. So the enzymes, not all enzymes work at 7. They really have evolved depending on where they're located in the body as to where they work and how they work. So that's the pH. The other big thing, though, is temperature. Just like if you fry an egg, you denature it. If you heat up or cool down an enzyme, that may slow or speed up how it functions. Typically, there is an optimal temperature for an enzyme. And for body enzymes, it's usually 37 degrees. If you raise the temperature, you might see the enzyme work faster, but eventually it would get hot enough so that that enzyme would actually denature, like the fried egg. So if it gets too hot, then you lose function because it's going to change the shape. If you chill it, that typically doesn't change the shape, but it takes away the energy available for the enzyme to work. And so cooling, like why you put food in the refrigerator, you're trying to slow the enzymes that are associated with bacterial and fungal growth, and that's going to help your food stay edible longer. If you just left it out on the counter at room temperature, it would start getting kind of funky within 24 hours. But you put it in the fridge, and it's good for 3 or 4 days. So lowering the temperature slows down enzymes. Raising the temperature might speed up an enzyme to a point, but then the enzyme will denature. It might lose its shape if the temperature goes above its optimal. And that's the whole premise behind why you keep cold foods cold and hot foods hot. Like if you go to a buffet, one of the things that your health inspectors do is they go through and measure the temperature of foods on the hot bar because they want to make sure that it is hot enough that any enzymes are denatured. And they check the temperature on things on the cold food bar because they want to make sure that it's cold enough to restrict growth of bacteria, molds, funguses. Because you don't want to end up transmitting anything. So that's that whole premise of hot things should stay hot, cold things should stay cold. Last topic. Enzymes can actually be affected by molecules called inhibitors. Now, if you just think of the word inhibitor, if you're going to inhibit, what are you going to do? You inhibit. You're trying to block it, right? So just think of an inhibitor. This is a blocker. So it's going to block enzyme activity. It's going to inhibit enzyme activity. There are reversible inhibitors. A reversible can block, but not permanently. It's only going to block the enzyme as long as it's there. Irreversible inhibitors create a permanent change. So reversible inhibitors, the inhibitor blocks the enzyme as long as it's there. But if you pull the inhibitor off, then you're going to have function come back. An irreversible inhibitor, that enzyme's done. It's not going to be able to function no matter what. So here is what they call a competitive inhibitor. So in this, E is enzyme, S is substrate, and do you notice that this is the normal reaction? Right? So normal reaction, you have an enzyme, it binds to a substrate, and you make a product. And then if there's more substrate, substrate to product, substrate to product, substrate to product. That's what the enzyme does. But a competitive inhibitor, where does it end up landing? If a competitive inhibitor comes into the reaction, what does it do to the enzyme? Where does it bind? It binds where the substrate should. So do you see how it's a blocker? Okay. Why it's competing? It's competing for that spot. So it binds to the active site where the substrate should bind. Yeah, so it's like the wrong key, right? So it slides into the spot where the right key should be, and so now the right key can't fit. So that's why they call it competitive, because it actually blocks the substrate. And as long as it's there, the substrate can't bind. This is different from a non-competitive inhibitor. So looking at this one, here again, this is normal. right? Going straight across the top. Enzyme plus substrate makes enzyme and product. Enzyme plus substrate makes enzyme and product. So the top row, that's the normal reaction. That's what the enzyme should do. But coming down, this is what happens when you add the inhibitor. So what happens here? If the inhibitor binds the enzyme, what does it do? Yep, it changes what? And it changes the shape of the active site, where the substrate should bind. So do you notice that it goes from being a triangle looking little piece to being like a little curved piece? So when the inhibitor is there, the active site is still there, but now suddenly it doesn't work. So instead of being like this, it's now like this. And so the substrate can't bind because it doesn't match the shape. So it changes the shape of the active site. And in doing that, it acts as a block for the reaction. As long as that inhibitor is there, then it can't. So that's a competitive versus non-competitive. So the last one is an irreversible inhibitor. So again, here's the normal, right? Straight across, enzyme plus substrate makes product. Enzyme plus substrate makes product. Enzyme plus substrate makes product, right? That's the norm. But then going down, what do you notice here? Instead of it going reversing, the arrow goes one direction. So when the inhibitor binds, that's permanent. So it does this permanently. It never comes out. And that means now this enzyme's done. So this enzyme can't function anymore because this is an irreversible inhibitor. So a lot of antibiotics are actually inhibitors. So they actually act to block activity of bacterial enzymes. So penicillin. So penicillin was actually found on accident. There was a guy, Alexander Fleming, was growing bacteria, or he wasn't growing bacteria, he was actually growing yeast. He was growing molds. So he had a plate that had a gelatin with some beef broth in it. So enough nutrients, like what you would use in microbiology, nutrients to allow for yeast cells to grow. So normally what you would do is you would take your yeast clump, spread it all over the plate, let it grow, and you would have a nice culture of yeast to be able to do his experiments. But what he found, so normally when you grow yeast, that grows all over the surface, nice and uniform. This is the normal yeast plate. So he did this, put it in to grow, came in, and instead he saw this. He saw these little spotty, clumpy, hairy looking areas, and no yeast grew around it. Well, actually, it's bacteria. But nothing would grow around it. There was like these open areas. So he said there has to be something that those cells are making that is inhibiting the growth around it. So it's an antibiotic because it blocks bacterial growth. So penicillin, they actually found this is what its chemical structure looks like. But what penicillin does, penicillin actually blocks or binds to the enzyme that makes bacterial cell walls. So a bacteria makes a cell wall. We don't make cell walls. But bacteria cell walls are pretty uniform looking, and they're nice and thick. And they're made by having proteins that are interlocking. So there's proteins and carbohydrates that all interlock, and it literally makes this look like chainmail. I think of like a chain link fence. But it's super thick on the surface of this little bacteria cell. And this prevents things from being able to attack a bacteria cell. So it makes this outer cover on the bacteria. But what penicillin does, penicillin blocks the enzyme from forming the links. So instead, what you end up with is you start off with like the little building blocks, but instead of having this chainmail, you end up with big holes in your wall. So this one gets linked, this one gets linked, this one gets linked, this one gets linked, this one, but not all of them. So over time, do you see that that's weak? That it's not reinforced? That it only has some of the links between? So this is what happens with penicillin. This is what happens without penicillin. And so literally the bacteria cell loses its cell wall, and that makes it very susceptible to attack. So macrophages in your immune system come along and go, hey, it's bacteria cell, and they'll like eat it and destroy it. Whereas with the cell wall, they don't see it. Like the cell wall is like cloaking. The cell wall like makes that bacteria cell almost invisible to your white blood cells. But if you can take that cell wall off, now underneath, the cell membrane gets picked up because it doesn't match what should be there. So that's really what penicillin does. Okay, so that finishes 10. So use this. So you've got 9 and 10. So like I said, you can go ahead and start making your list of things that you're going to want to add to your